In this video, we will discuss about the PDAC progression in terms of glycan or aberrant glycosylation that are occurred in the pancreatic cancers. And later on, we will discuss how this aberrant glycosylation also helps in the immune evasion. So let us quickly understand these topics in brief. So this is the normal pancreas. So when because of the mutations within this normal pancreas, it might and over several period of time it might leads to the generation of tumors in this normal pancreas, and during this uh, period of generation of the tumors, there is a changes in the glycosylation profile that has been observed also. That initially, so what will happen is that the truncation of O glycans will begins when the normal pancreas starts to move towards pancreatic starts to move towards the abnormal or pancreas uh, with a tumor. So initially the truncation of O glycans will be occurred, and thereafter the aberrant salic acid at the aberrant salic acid formations at the cell surface will be observed. And when it further moves towards the pancreatic cancers tumor stage, then Lewis antigens will start to form, such as TN antigen, STN antigen, or T antigens. And thereafter when it progresses further, it will leads to the mucin formation. Mucins is the O glycan long you can say glycosaminoglycans type structures that are formed at the cell surface. And after that, the TME lectins will be formed, or we can say that lectins which are affected by tumor microenvironment. So how this changes in glycan plays a role in the metastasis is shown in the bottom figure, where the tumors with the aberrant glycosylation will be able to extravasate into the blood vessels with the help of with the help of this we can aberrant glycosylation profile. Though it is not shown here, but we have already covered how this aberrant glycosylation helps in the metastasis. You can go through those videos to understand these things. And after that, when it reaches at the when it reaches at other sites, or when it reaches at the secondary organs where it needs to infect, then based on this aberrant based on the Lewis antigens that are present on the tumor cells, it will be able to interact with the E-selectin, and that will helps in the slow down of this tumor cells, and thereafter it helps in the intravasation of these tumor cells at the secondary organ, and there it can metastasize or spread the tumors. So this is in brief how the aberrant glycosylation helps in the intravasation from its primary organ sites to the extravasations from its primary organ sites to the intravasation at its secondary organ sites, and that helps in the spread of the tumors across different part of the body, or we can say metastasis. And how this aberrant glycosylation also helps in the immune evasions is shown in this figure, and it is briefly categorized into four categories. First one is the elongated protein formations, glycoprotein formations, and because of this it will act as a physical barrier, and some people even call this as a glycocalyx layer. So this will helps in preventing this direct interactions of this immune cells with the cancer cells. Thereby they will not be this prevention is mainly based on steric hindrance, that it will act as a physical barrier. Thereby the immune system will not be able to generate response against the cancerous cells. It will not be able to detect those uh, unique antigens on the cancerous cells, or non-self epitopes on the cancerous cells. Next thing is that because of the overexpression of the salic acids, because of the overexpression of salic acid at the cell surface, because of the upregulations of this ST3 GAL1 and GAL4, these salic acids can be act as a ligand for the CLX7 and CLX9 receptors that are found on the monocytes. And these interactions helps in the differentiation of the monocytes towards M2 phase or M2 phenotype, which will act as a tumor associated macrophage. It will helps in the tumor progression, and also prevent the tumor cells from immune attack. And we have already covered these things in detail in one of our previous videos. You can check the I links or go through the reference or uh, go through our video playlist to understand this topic in detail. And next thing is that the overexpression of the mucins in the tumor cells because of the mutations will release some inflammatory cytokines, and that helps in the upregulations of this myeloid derived suppressor cell and the regulatory T cells, and this will then helps in the suppression of the activated T cells and reduce the immune response. The next thing is kind of reverse that of step 3. In this case, the tumor microenvironment that has been formed will release some cytokines that will lead to the upregulations of the ST3 gal4 FAT1 FAT1 2 6, and this will lead to the overexpression of the we can say overexpression of this Lewis antigen such as S Lewis X, S Lewis Y, and this can interact with the receptors that are present on the immune cells and suppress the immune response against and helps in suppressing the immune response against the cancerous cells. So this is the 4 main category by which this PDAC cancerous tumor cells helps themselves in immune evasion. If you need to understand about this topics in detail, you can go through the reference or our previous videos. A reference link is provided in the description, and some of the previous videos links are provided in the I button. So that's all for this video. Thank you for your time.